Hello everyone, Ramya, Assistant Professor of CSC Department from Rohini College of Engineering and Technology. Subject code OEC 754 Unit 5. Topic Endomicroscopy. Objective to learn about the concepts of endomicroscopy and its types. Endomicroscopy is a technique for obtaining histology like images from inside the human body in real time, a process known as optical biopsy. It is generally refers to the fluorescence confocal microscopy, although multiphoton microscopy and optical coherence tomography also been adapted for endoscopic use. Here endomicroscopes either have a miniaturized scanning head at the distal tip of the imaging probe or perform the scanning outside of the patient and use an imaging fiber bundle to transfer the scan pattern to the tissue. Here some of the types of endomicroscopy: single fiber endomicroscopes, fiber bundle microscopes, distal scanning endomicroscope, non-focal endomicroscopes. First we are going to see the single fiber endomicroscopes. Single fiber confocal endomicroscopes use the tip of an optical fiber as a spatial filter. It is enabling miniaturization of the microscope. Here we have 488 nm blue laser passes from the source through an optical fiber to a flexible. An image is generated by scanning the focused spot throughout the image plane and compiling the point intensity measurements. The image plane can be translated up and down in the sample, allowing generation of 3D image stacks. Next one is fiber bundle endomicroscopes. Here fiber bundles are originally developed for use in flexible endoscopes and have since been adapted for use in endomicroscopy. Here it also consists of a large number of fiber cores inside a single shade cladding or flexible and have diameters on the order of a millimeter. In a coherent fiber bundle, the relative positions of the cores are maintained along the fiber. It means that an image projected onto one end of the bundle will be transferred to the other end without scrubbing. Here also, if we have one end of the bundle is placed at the focus of a tabletop confocal microscope, means then the bundle will act as a flexible extension and allow endoscopic operation. Here each core essentially acts as an image pixel, and so the spacing between fiber cores limit the resolution. In addition of micro optics at the distal tip of the bundle allows for magnification and hence higher resolution imaging by the cost of reducing the field of view. The next one is digital scanning endomicroscopes. It is used to incorporate a miniature 2D scanning apertures into the imaging process, and also the laser excitation and returning fluorescent emission are sent to and received from the scanning head using an optical fiber. Here the most experimental devices have either used MEMS scanning mirrors or direct translation of the fiber using electromagnetic acquisition. The next one we are going to see non-confocal endomicroscopes. Here wide field endomicroscopes have been developed for selected applications including the imaging of cells. Here optical coherence tomography and multiphoton microscopy have been demonstrated endoscopically. Here we have some of the applications. First one is endomicroscopy has been used to study many gastrointestinal disorders. Small bowel disorders investigated with endomicroscopic include celiac disease and graft versus host disease. Here gastric cancer and Helicobacter pylori gastric cancer have been imaged with endomicroscopes. Then the endomicroscopes has been used to help target biopsies in all these disorders as well, and may also reduce the number of biopsies needed to achieve diagnosis. The next one is confocal endomicroscopy. It is recently developed endoscopic technology that allows for histological analysis of tissue in vivo. That means conventional endoscopy involves identifying lesions grossly followed by biopsy for histological analysis. It also allows for the performance of real-time biopsy during endoscopy by observation of the muscular layer of the gastrointestinal tract of the cellular. Here also we have two systems are currently available. First one is tip integrated confocal laser endoscope and a flexible fiber based confocal mini probe. That means the technique of confocal endomicroscope has been used for diagnosis of upper gastrointestinal disorders such as Barrett's esophagus, gastric carcinoma, celiac disease. Next one is technical overview, basic principle of confocal microscopy. Here the CLE is based on low power blue laser light based tissue exposure and fluorescence. That means the laser light is focused on the area of interest and backscattered light is then focused onto the detection system by the lens. Here then the backscattered light passes through a pinhole apertures which increases the resolution of the image. Here also a dye is used to provide the contrast for adequate visualization and serial optical sections are obtained oriented parallel to the tissue surface. The next one is probe based CLE. Here it is the probe based CLE system comprises a fiber optic bundle with an integrated distal lens that is connected to a laser scanning unit. Here also the probe based system to date has a fixed focal length and so it can only scan in a single plane unlike current microscope systems that can create cross-sectional images at different depth. Here also we can use the individual optical fibers function as the pin core. It is designed for use during ERCP, require an endoscope accessory channel of at least 1.0 mm, whereas the other probes designed for use in EEGD and colonoscopy require a channel of at least 2.8 mm. All probes generate dynamic images. Here the depth of imaging from the surface of the confocal lens is 40 to 70 mm for Chollinger flex probes and 55 to 65 mm for both gastroflex UHD and colorflex UHD probes. Here we have the resolution is 3.5 mm, whereas for gastroflex UHD and colorflex UHD probes it is 1 mm, so that the probes can be refused after the disinfection for as many as 10 to 20 examinations. The next one is endoscope based CLE. Here the diameter of the endoscope is 12.8 mm and the tip length is increased to accommodate the laser microscope so that there is a 5 cm rigid portion. It can be used for upper and lower GI tract examinations but is too large for pancreatic imaging. Here the images are collected at a scan rate of 1.6 frames per second or 0.8 frames per second with an adjustable depth of scanning ranging from 0 to 250 mm as a field of view of 475 mm. Thank you.